How to use today in Flippin Science? We're looking at factors affecting exchange. Thanks to Sam Moyle for producing this PowerPoint. So science understanding is we're going to look at, explain how the exchange of materials across membranes is affected by factors including surface area to volume ratio of the cell, concentration gradients, and the physical and chemical nature of the materials being exchanged. So let's go into those factors. There's several factors that will affect the rate of exchange of materials between the cell and its environment, so from the intracellular space to the extracellular space and vice versa. Surface area to volume ratio explains the size of cells. So cells are small because that maximizes surface area to volume ratio, so you get a fast exchange between the cell and the environment. Concentration gradients compare the concentrations within the cell to the concentration in the extracellular space. The higher the concentration gradient, the faster the movement. And the nature of the exchange materials determines how easily they can pass through the membrane in terms of things like size and charge. So let's have a look at those. So let's talk about surface area to volume ratio. It impacts our lives a lot, but we often don't think about it. When you're cooking, you often cut up your food because it takes less time to cook. When you're cooking, you're heating up food and you need to get heat energy from the outside of the food into the middle of the food so it's fully cooked. If you cut it up into smaller bits, there's much more area exposed from the surface for heat energy to make its way into the food. If you think of how long it would take to cook fries versus an entire potato, that gives you an idea of how chopping speeds things up. If you're going to have a party, you get a bag of ice, you smash that ice before you chuck it in the esky, because then that breaks up one big block of ice with a certain surface area into lots of small pieces of ice with larger surface area. So there's more surface area exposed to the environment, which means you'll be drawing more heat energy out of your drinks, so they'll cool down a lot faster. So that's surface area to volume ratio. We're making things smaller that increases their surface area compared to their volume. And this applies in biology. So as a cell increases in size, its surface area to volume ratio decreases. That means there is less surface area exposed per unit volume. And that means there's less surface area compared to the volume for exchange to occur over, and cells need stuff from the extracellular space, and they also need to give stuff back to the extracellular space. So they need to get nutrients in, and they need to get waste products out. And if you have too small a surface area to volume ratio, you can't do that very efficiently, and that can be very bad for the cell. So if a cell has too low a surface area to volume ratio, the efficiency of exchanging materials with the extracellular space is decreased, and in fact it's a trigger for cell division when cells get too large. So let's look at this in maths. So let's say we have a cell that's 1 millimeter in cell radius, which is absolutely huge for a cell. The volume of that cell would be 4/3 of pi, which is 1.5 pi's, and the surface area would be 4. So 4 divided by 4/3 is 3, so it's a 3:1 ratio in terms of surface area compared to the volume. If we double that cell's radius, so now it's a 2 millimeter radius cell, which again is absolutely huge for a cell. Our volume has gone up to 32/3 pi, and our surface area is only 16 pi. 16 divided by 32/3 gives you 1.5, so it's 1.5 to 1. What that means is the surface area has increased by a factor of 4, but the volume has also increased at a much larger ratio, so that means we have less surface area compared to the volume that the cell is now containing. And to finish off this idea, if we triple the radius of the cell, our volume is 108 on 3 pi, our surface area is 36 pi. We can see again the surface area has increased, but the volume has increased by a much larger proportion. So now our ratio is 1 to 1. 108 divided by 3 is 36. So what that means is as our cell is getting bigger, it's having proportionally less surface area compared to the volume of the cell. And that means it's much harder to get things like oxygen in, carbon dioxide out, glucose in, urea out. All of those things become a lot more difficult when you have less surface area exposed to the environment for the volume that you're containing. We can also do this with cubes rather than using circles over here. So if we look at our, we have a 1 unit cell here. Surface area is length times width times number of sides. That gives us unit 6. That gives us 6 units squared of surface area. Volume is 1 by 1 by 1. So our surface area to volume is 6 to 1. Instead of a 1 by 1 by 1 cube, we'll make it a 5 by 5 by 5 cube. The surface area is bigger because we're making a bigger cube, but the volume is much bigger proportionally than the smaller cube. We're almost at 1 to 1. So it's 1.2 to 1. So we have a much smaller surface area compared to the volume that we're containing. But if we were to cut up our big 5 by 5 by 5 cube into lots of 1 by 1 by 1 cubes, we would end up with the same ratio that we started with. So what that means is if we have lots of small things that have a large surface area compared to the volume that they're containing, and that means in terms of cells it's much easier to exchange materials with the environment. Let's talk about concentration gradients now. So in diffusion and osmosis, particles move with the concentration gradient from high concentration of solute to low concentration of solute in diffusion, and high concentration of solvent to low concentration of solvent in osmosis. We've looked at diffusion and osmosis before. Greater the difference in concentration, the faster the movement across the cell membrane, and that's just down to thermal movement of particles. So when we talk about concentration gradient, we're comparing one side of the membrane to the other. So if you have a much higher concentration on one side compared to a low concentration on the other, that's a steep concentration gradient. There's a large amount of difference in concentration from one side of the membrane to the other, so you get a fast rate of diffusion. Whereas if the concentrations are fairly similar, we've got a less steep concentration gradient, it'll be a much slower rate of diffusion. Fewer particles will pass across the membrane per unit time if the concentrations are fairly similar from one side to the other. And this applies to osmosis as well. In osmosis, rather than looking at solute moving from one side of the membrane to the other, we'll be looking at water moving from one side of the membrane to the other. Now, a factor affecting exchange is active transport. If you want to move something against the concentration gradient, you need to put in energy to do that. And we've looked at this in terms of membrane proteins already, but we'll just cover it again. So active transport moves substances against the concentration gradient, and this can be done by pumping. So we have a high concentration of substance outside the cell, we've got a low concentration inside. If we're trying to keep this concentration lower inside, we would be pumping actively that substance out. We'd be using energy to keep that substance out of the cell, because it will just diffuse back in, depending on the nature of the substance. This is active transport. We're moving against the concentration gradient, and sometimes just moving large things, as we'll see, requires energy too. So what is being exchanged is also important. So small uncharged particles like oxygen and carbon dioxide, they can pass through the membrane relatively easily, not particularly quickly, but relatively easily via diffusion. The fact that they're uncharged, and in fact these are non-polar, means that they pass through relatively easily through the membranes. Something like water, however, which is a polar molecule, has a bit more difficulty. That's why we have those membrane protein channels that can move water across the membrane a bit faster. Large particles, so glucose is our example of a pretty large particle, or a charged particle like an ion, like a sodium ion, or in this case we've got a hydrogen carbonate ion down here. They may require assistance from channel proteins, carrier proteins, or endo and exocytosis. So these processes can move those substances across the membrane, but they may require energy to do that. So channel proteins don't require energy, they just make it slightly easier to move things across. Carrier proteins, depending on the way they work, they might be just a facilitated diffusion, which doesn't require energy of the cell, or it might be an active transport protein that will require energy from the cell. And endo and exocytosis is when we're manipulating the cell membrane or vesicles within the cell to move substances into or out of the cell, and that requires energy because you're moving the cytoskeleton to do those things. So the type of material that you're exchanging in terms of things like size or charge will determine how that substance moves across the membrane. So let's just quickly look at those. So here's a protein channel, so it's a channel in the membrane. It's got a hole that's just wide enough for that substance to pass through, so that doesn't require energy from the cell to do it, but it will make what is essentially diffusion faster, or in case of a water channel it would be osmosis faster. Carrier protein, this could be facilitated diffusion, so as the particle moves into the protein changes shape and then that spits it from one side of the membrane into the other. That might not require energy, but depending on what the substance is, it might require energy. And we have our endo and exocytosis. So endocytosis, we're making a pocket in the cell membrane. This is done by manipulating the cytoskeleton around the membrane in order to make that invagination, and then the substance is taken into the cell. Exocytosis is the opposite. We've got a vesicle inside the cell that's being pushed by the cytoplasm to the cell membrane, and then that opens up and that releases the substances out. This will require energy, and this will require energy as well. So the key thing is the type of substance will determine how it is transported across the membrane. So let's do our check. Explain how the exchange of materials across membranes is affected by factors including surface area volume ratio of the cell, concentration gradients, and the physical and chemical nature of the materials being exchanged. So today on Flippin Science, we looked at factors that affect exchange. That's it for Flippin Science today. See ya.